Hi friends, today we are going to discussing about one important and basic concept in immunology. That is, what are the different types of cells involved in immune system? So if you take immunity, what is immunity? It is a body defense or protection against any infections. So any living organism shows defense or protection against infections. This is called immunity. What is immune system? The group of cells that are involved in immunity process is called immune system. So the total cells that are involved in immune or immune function are called immune system. Immunity or immune system generally found in all living organisms, starting from bacteria to higher organisms, but it is well characterized in vertebrates. Vertebrates means from fishes to humans, so mammals. So majorly characterized in vertebrates, but it is present in all living organisms. Blood and blood components perform immunity functions in vertebrates. So vertebrates, all vertebrates contain blood. So blood and blood component majorly perform immunity function. Because of the blood presence, immunity or immune system exists. So blood and blood components perform immunity function in higher organisms. What are the different types of immunity? Immunity broadly categorized into two types. Those are innate immunity and acquired immunity. Innate immunity is also called natural immunity. It comes by birth. Again, this innate immunity categorized into three types. That is specific immunity or specific immunity. That means one species have one immunity to one particular disease, another species has another type of diseases. Best example is HIV. So HIV is more prone to humans, whereas monkeys are not prone to HIV. That means one species is more prone to one particular disease, but the another species not prone to that particular disease. Best example is HIV. HIV is so humans are susceptible to HIV, whereas monkeys are not. This is best example for specific immunity. Another one is racial immunity. Racial means, for example, Indians and Africans. These are two different races. Both are humans, but these are different races. One race has immunity to one particular disease, whereas another not. For example, we are not much prone to malaria, whereas African people are very much prone to malaria. This is called racial immunity. That means within the species, different races present. One race is susceptible to one particular disease, whereas another race is not. And the third one is individual immunity. So each and every person has its own immunity, an individual immunity. That individual immunity is known as personal immunity or individual immunity. Very best example where this is mainly due to genetic make. So these are the three different types of innate immunities, that is specific immunity, racial immunity, individual immunity. Another type is acquired immunity. Acquired immunity means any living organism during his lifetime it acquired immunity. This is known as acquired immunity or artificial immunity. This is also called artificial immunity. And this acquired immunity again categorized into active and passive. If you take active, these again two types. Natural active immunity. Natural active means through clinical or subclinical infections. Whenever one infection taking place to the organism, subsequently it get resistant to that infection. For example, smallpox, chickenpox, like that. So if whenever a person come in contact with the smallpox virus, once he recovered, then in his lifetime he won't get that particular disease. This is called natural active immunity. Another one is artificial active immunity. Best example is vaccination. We are giving vaccine. Vaccine is what is vaccine? Vaccine is a microbial preparation, and we are giving that vaccine to the particular organism. And in future, that person, vaccinated person, won't get disease to a particular organism. This is called artificial active immunity. So these are two types of active immunity: natural active immunity and artificial active immunity. Natural active immunity means through infections, whereas artificial active immunity through vaccination. Another one is passive immunity. Passive means again two types: natural passive immunity, artificial passive immunity. Natural passive immunity, transplantal IgG. For example, when baby is in mother womb, through placenta, IgG transform from mother to baby. Similarly, through breast milk, IgA transferred from mother to baby. So in this way, the baby gets immunized. So this is called natural passive immunity. Another one is artificial passive immunity, anti-serum. Whenever one person is recovered from a particular disease, From that person, we take serum and inject it into diseased person. This is called artificial passive immunity. In this way, immunity is categorized into innate immunity and acquired immunity. Innate immunity again categorized into specific immunity, racial immunity, and individual immunity. Whereas acquired immunity categorized into active and passive. Active means natural active, artificial active. Natural active means through infections, whereas artificial active by vaccination. Passive immunity, natural passive, artificial passive. Natural passive, transplacental IgG transfer or transfer of IgA from mother to baby by breast milk. Similarly, artificial passive anti-serum. That means whenever one person get uh, recovered from the particular disease, take blood or serum from that recovered person and inject it to another diseased person. That is called anti-serum injection. This is the different types of immunities. And what are the different types of cells involved in immunity? So these are all different types of cells involved in immunity. Whatever the cell, as I said earlier, blood majorly involved in the immune system in vertebrates. So, blood developed from one particular stem cell that is called hematopoietic stem cell in bone marrow. So, origin of blood is bone marrow, and in bone marrow, the origin cell or pioneer cell for the blood cells is hematopoietic stem cell. This hematopoietic stem cell again divided into two types. One is myeloid progenitor cell, another one is lymphoid progenitor cell. So hematopoietic stem cell is the uh, pioneer cell for the blood. It is present in bone marrow and divided into two types of cells. One is myeloid progenitor cell, another one is lymphoid progenitor cell. After dividing as myeloid progenitor cell and lymphoid progenitor cell, myeloid progenitor cell develop into other types of cells. Similarly, lymphoid progenitor cells also develop into another types of cells. First, if we take myeloid progenitor cell, these myeloid progenitor cells further divided into granulocytes and monocytes. Myeloid progenitor cell further developed into granulocytes and monocytes. Granulocytes, you all know granulocytes are acidophils, basophils, and neutrophils, and monocytes. So, granulocytes and monocytes are developed from myeloid progenitor cell. And different types of granulocytes are, first one is basophils. This is the basophils. It contains bilobe nucleus. So, basophil, what is the function of basophils in immunity means they regulate allergic reaction. Whatever the allergic reactions or anaphylactic reactions are there, all these anaphylactic or allergic reactions are mediated by basophils. And second one is eosinophils or acidophils. These eosinophils or acidophils also develop from myeloid progenitor cell. They are involved in defense against nematodes. Whatever the nematodes infections are there, the nematode infection is uh, protected from nematode infection. We can protect it by eosinophils and other parasites also. Eosinophils involved. So defense against nematodes and other parasites performed by eosinophils. And third one is neutrophils. These are the first cell respond against infection. Whenever infection taking place to the body, the first cell respond is neutrophil. So neutrophil is the first cell which respond to the against infection in our body. Like mast cells. These are the another type of myeloid cells developed from myeloid progenitor cells. And these mast cells involved in control immune system function by producing histamine, heparin, cytokines, and growth factors. So all these things are produced by mast cells. So it controls immune system function by producing histamine, heparin, cytokines, and other growth factors. Some studies showing that mast cells also derived from basophils. In some books, we can see that mast cells are derived from basophils. So these four types of cells are called granulocytes: basophils, eosinophils, neutrophils, and mast cells. All these granulocytes are derived from myeloid progenitor cell. And another type of cells that are derived from myeloid progenitors are monocytes. So the monocytes again divided into macrophages and dendritic cells. So another type of non-granulocytic cells that are derived from myeloid progenitor cells are monocytes, and these monocytes again divided into macrophages and dendritic cells. Macrophages and dendritic cells are popularly called as antigen presenting cells. Antigen presenting cells means whenever any foreign pathogen or foreign infectious agent enter into body, that infectious agent or bacteria or virus is taken up by these antigen presenting cell, that is macrophage and dendritic cell, and present to the immune system. That's why they are called antigen presenting cells. So macrophages and dendritic cells are popularly called as antigen presenting cells, and these are derived from monocytes. And if you take macrophages, the macrophages travel through blood and settled in particular tissue and named different names are given. For example, if macrophages settled at bones, then that macrophages are called osteoclasts. If macrophages settled in the bone, then that macrophages are called osteoclasts. Whereas some macrophages settled in lung, then that macrophages are called alveolar macrophages. Some settled in brain, that macrophages are called microglial cells. Some settled in connective tissue, that macrophages are called histiocytes. Some settled in liver, they are called Kupffer cells. Some macrophages settled in skin, called Langerhans cells. So these are the different types of macrophages settled in different tissues, but the function is same, that is antigen presentation. So whenever these macrophages settled in particular type of tissue, the name is different there. So in this way, different types of macrophages are there. Dendritic cells are only one type, and these dendritic cells also involved in antigen presentation. So these are the different types of cells derived from myeloid progenitor cells. So myeloid progenitor cells further divided into granulocytes and monocytes. Granulocytes are basophils, eosinophils, neutrophils, and mast cells, whereas monocytes are monocytes, and these monocytes further divided into macrophages and dendritic cells. Macrophages and dendritic cell popularly called as antigen presenting cells. Based on the tissue localization, macrophages again named as different types, but the function is same, that is antigen presentation. This is about myeloid progenitor cells. And another progenitor cells are lymphoid progenitor cells. All lymphocytes coming from this progenitor cell. These lymphoid progenitor cell further divided into three categories. One is T cell progenitors. T cell progenitors means T lymphocytes. They give T different types of T lymphocytes. Because why they are called T cells means after generation of lymphoid progenitor, some of the cells transfer to the thymus and they grow and develop there. So because these lymphoid cells developed in the thymus, the name given as T cell progenitors. These T cell progenitors again divided into memory cells, cytotoxic T cells, and helper T cells. So these three are different types of T cells, that is memory T cells, cytotoxic T cells, helper T cells. Memory T cells involved in remembering the foreign virus particles. Generally, T cells majorly involved in removal of viruses and cancer cells. T cells are mainly involved in removal of virus and cancer cells. And T cells involved in cell mediated immunity. So cell mediated immunity, they involved in cell mediated immunity. T cells involved in cell mediated immunity. In cell mediated immunity, they are divided into three types. One is memory T cells. They remember the antigen or virus present enter into the body. And second one is cytotoxic T cells. Whatever the cell infected to the virus or cancer cell, they destroy that infected cell. That is called cytotoxic T cell. And another one is helper T cell. This helper T cell involved in both facilitate the development of or execution of both cell mediated immunity and humoral mediated immunity. Helper T cells helps the uh, execution of cell mediated immunity and also humoral mediated immunity. These are the helper T cells. Generally, these helper T cells recognize the antigen presenting cells and tell to the T cells and B cells and removal of the antigen process. So to help T cells involved in both cell mediated immunity and humoral mediated immunity. So these are the different types of T cells. And another type is B cells. They are developed in bone marrow only. That's why they are called B cells. And these B cells again categorized into plasma cells and memory cells. Memory cells involved in remembering the antigen or bacteria, whereas plasma cells involved in the production of antibodies. Generally, B cells recognizes bacterial pathogens, whereas T cells recognize viral pathogens. So, if viral pathogen enter into body, T cells respond, whereas bacterial pathogen enter into the body, B cells respond. B cells are two types. Memory B cells, plasma B cells. Memory B cells remembering the bacterial pathogen, whereas plasma B cells identify the pathogen, bacterial pathogen, and produce antibodies against to the particular bacterial pathogen. So these are the different types of lymphoid cells, that is T cells and B cells. T cells involved in cell-mediated immunity, whereas B cells involved in humoral-mediated immunity. What are the cells that are helped in both type of immunity, both T cell-mediated, cell-mediated immunity and humoral-mediated immunity means helper T cells. So helper T cells belongs to both cell-mediated immunity and humoral-mediated immunity. And another thing we remembered is B cells sometimes also act as a antigen-presenting cells. What are the different types of antigen-presenting cells? Macrophages, dendritic cells, and B cells. And another question is which type of cells involved in both cell-mediated immunity and humoral-mediated immunity means helper T cells. So these are the different types of cells involved in immune system. If you take the immune process, whenever foreign pathogen or virus enter into body, these foreign pathogen or virus taken up to the antigen presenting cells. That is one of the antigen presenting, that is macrophages, called phagocytosis. Once the pathogen enter into body, that is phagocytosis happen, then this pathogen break down into small pieces and presented on the surface of the macrophage or antigen presenting cell. These presented antigen particles recognized by the T helper cell. As I said earlier, these T helper cells involved in recognition of foreign pathogen or antigen and tell to the T cells or B cells. So once our recognition of the pathogen or pathogen particles by the T helper cells, they either tell to T cells or B cell. If it is viral pathogen, it tell to T cells. If it is bacterial pathogen, it tell to B lymphocytes. If it is viral pathogen, they tell to T cells, and these T cells involved in cytotoxicity or killing of the particular infected cell. And finally, these cell particles are phagocytosed by the macrophages and removal of that infected cells. If the infected pathogen is bacteria, then B lymphocytes respond. They produce plasma cells and memory cells. These plasma cells again produce antibodies. These antibodies attach to specifically bind to the particular pathogen, and these are removed by complement system finally. So in this way, the whenever antigen enter into body, the antigen presenting cells present the antigen on the surface. These are recognized by the T helper cells. After recognition, based on the type of pathogen, whether it is viral pathogen or bacterial pathogen, they removed either by cell-mediated immunity. If it is viral pathogen, that is removed by cell-mediated immunity. If it is bacterial pathogen, that is removed by humoral-mediated immunity. And clearance of the antigen taking place. This is all about immune process and different types of cells involved in the immune system. Thank you.